So hi, hello, and welcome again, Microbehunter here, and welcome to a somewhat unusual time for live stream. Yeah, it's a Sunday, at least where I live, and I already did a live stream yesterday, 12 hours ago, approximately. And I'm going to do today another live stream on a very similar topic, and the reason is because I would like to see which time is more convenient for you. So I'm going to look at the statistics, and then maybe because of the time difference there, I got the information that some people were not able to watch yesterday. So I'm going to try, to offer now also a second time, and we're going to see which one is going to be more convenient. Today, I'm just like yesterday, I would like to put some water fleas under the microscope, and yesterday I added a little bit of caffeine to those water fleas to observe the heartbeat and what effect caffeine has on the heartbeat, on the heart rate of those water fleas, which you see over here. Today I would like to add a little bit of yeast as a source of food, because then we can see the water streaming. So a little bit some organizational things. The live stream will basically last maximum 1 hour. It depends a little bit on how many questions I get, and I assume that maybe there will be new people joining in the live stream because of a different time. So different people might be able to join in. If you're totally new to the live stream, it has become tradition that if you go to the chat, then maybe you can quickly tell me from where you are from, and to also please give me a short note if you're able to hear me well, because sometimes the sound is not fine. And normally during the live stream, as I go on, I will often interrupt myself a little bit, and I will read off the comments and answer some of the questions that you may have. Okay, so I'm going to read all of the comments, but I might not be able to respond to all of them. Okay, so there is a good afternoon from the Philippines. Say hello, Sarah. Okay. How about feeding and giving him some alcohol to slow down the heartbeat and overall metabolism? This is something that I can actually try. Okay, maybe we can see him eat in slow motion. Well, we're going to see about that. In this, alcohol indeed is able to slow down the heartbeat, and caffeine does speed it up. We can actually also experiment with that. And it's now easier to watch a live stream because there it's 3 o'clock in the afternoon. Okay, so hello from Thailand. And hello from Denmark, from Australia, from Norway, the sound is good. Okay. If you have two different microscopes, each viewing red blood cells, and they both have the same magnification, and will one microscope's image make the cells appear larger than the other at the same magnification? No. At the same magnification, it should not appear different. What might appear different is the field of view. So basically how much you're able to see, right? So and this depends on the eyepieces, but otherwise the magnification itself should be the same. But if the eyepieces have a small field of view, then it looks a little bit like looking more through a little bit like like a keyhole or something. Okay. Greetings from Lower Austria. Yes. Hello. Greetings from Upper Austria. I have to say here, excellent audio and video. So so what we have over here is, of course, a water flea, and I picked it up yesterday, and I'm going to now show you very quickly how I collected the sample. So this is now a video. So this is about a couple of minutes from where I live. It's a little, it doesn't play. Okay, now it plays. Okay, I got stuck a little bit. Let's start again. Yeah, so here we go. So that's basically in the forest, and there is a little puddle with water. Generally, the puddle should be at least several days, if not a couple of weeks old. So this one here is maybe already 2 or 3 months old. The one on the left side already dried out. And this gives it a sufficient time for water microorganisms to grow. There are lots of insects there as well. And this is where collected now, simply using a jar. I collected some of the water. It was quite muddy, so it's kind of difficult to see all of the water crustaceans and the living things that are in there. However, if you allow the jar to stand for a day or so, then all of the suspended particles will settle down to the bottom of the jar. jar, and then it will also be much easier to find them. And yeah, and you maybe are able to find see those little water crustaceans. There were several ostracods in there, and also water fleas, Daphnia, and others as well. So what I've done is I basically transferred them using a pipette into a petri dish here, because it's simply much easier to pick them up. And then I've already transferred them to a microscope slide, and I did not add any cover glass. So what you can do quite easily do is you simply go in here and look, I cut off the tip. This is a disposable pipette, plastic pipette, and I simply used some scissors to cut it off because otherwise it's too narrow to pick up any larger water organisms. So you just go in here. No, I didn't catch that one. So you go in here, you quickly suck it up. You transfer it over here to the slide. You might be able to see the little one over here. And what I usually do is I will remove any excess water again. And then there is a, you might be able to see it better now here. Looks like an ostracod here. Then basically no cover glass, and then it's not able to swim away. So, and this is actually the way that I usually observe them. And under the microscope, it looks like this. Now you're going to see all of those rainbow colors. This is because the surface of the water is not smooth because of the surface tension, and I'm using polarized light and a whole bunch of filters. So under bright field, this is how you probably are going to see it. So these are now the natural colors. Okay, so what I'm going to do now, as always, before I start feeding them, and there's a reason why I do that, because we can see it in the water movement. I always go back a little bit every now and then back into the forum, into the chat forum, to read some of the questions here. Okay, good morning from midnight. In California, it's midnight right now. Okay, now it's easier to watch a live stream because it's 3 in the afternoon. From India, from Thailand, from Denmark, from Australia. I'm kind of surprised, even though it's at a different time, there's still a lot of people joining in. From Lower Austria, already read that. What is your favorite microorganism is the question. Well, I don't have a favorite one really. But generally, I can tell you which ones are less favorite ones. And the less favorite microorganisms are, believe it or not, bacteria. Even though at university I studied bacteria, I have a training as a bacteriologist. I consider them, under the microscope at least, relatively not so interesting bacteria, because they're quite small, difficult to see. Some of them are more interesting, sure. But I think the larger organisms are much more fascinating to observe. And I'm saying that because many beginning microscopists, the first thing that they want to look at are bacteria. And I think that they are much more interesting specimens to look at. So hello from Pakistan, from Australia, and better timing now when the daylight saving ended and the live streams were too early in the morning. Okay. From Barcelona, New Jersey. Make sure that the eyepieces have the same magnifications too. Yes, that's of course correct. That's something that I forgot. The eyepieces have to have the same magnification. So it was a question if you have different microscopes and the magnification is the same of the objectives. Does it look then the same? Okay. Hello from India. Is it possible to see cell organelles under the microscope? Yes, not all of them, but several of them. So there are certain cell organelles that you can only look at using an electron microscope, but especially when we look at paramecia, for example, then some cell organelles can be seen quite well. You deal with a common hashtag for your microscope videos, maybe compiled in a playlist of your channel mentioned yesterday is excellent. Okay, this is another recommendation that I have, which I kind of floated yesterday in the live stream about 12 hours ago, is the following. I've been playing with the idea of actually doing something a little bit like a virtual, how do you call this, hobby microscopy conference. Normally in a conference you have talks and speeches and so on. And I thought maybe I'm able to motivate other people who want to make videos and to share their knowledge. And you simply upload a video or videos on your YouTube channel. And even if it's not a separately dedicated channel for microscopy alone, but if you do some kind of a hashtag, then maybe it's much easier to actually find those videos. And I was kind of thinking maybe to have something like a virtual conference where we're able to share a little bit of knowledge. So I've been floating this idea yesterday, and What's a cheap but good microscope you could recommend? I'm looking for another microscope to add to my collection. Oh, this is a very large and complex question. What is a cheap and good microscope? Because what is good depends quite a bit on the person. Okay, different people have very different opinions of what is quality. And for this reason, I do not really want to give a recommendation, but I did make several, how do you call this, review videos on my other channel. So this here, this little bean-shaped organism is called an ostracod. It's not moving right now. It's moving a little bit. Okay. Sometimes it doesn't move because there is already too little water. Yeah. And let's have another one over here. Look at another one over here. Where is it? Yeah, here. That's another ostracod. Yeah. So these are also water crustaceans. So they're yeah. Can also see that I think they're not quite as easy to look at as like for example Daphnia, because they're not quite as transparent. Now, yeah, here let's have a look at here, because what we're able to see here on the back are two things. Let me turn on the arrow again. So this round structure, that is an egg. Okay, sometimes some of those water fleas actually they have several of those. And this here is the heart that's beating here, right? And another thing, and we were actually able to see this here much better than in the one that I had yesterday, because there are less algae growing on this water flea here. You can also see the eye here, and you might be able to see the muscle fibers or the that actually pull on the eye to make it move around. So what I'm going to do now is the following. I'm going to now prepare a little bit of food. Because I would like to add some yeast because this allows me to see now the streaming of the water, because yeast cells being significantly smaller, they are floating around in the water, and this way we're able to see a little bit the water stream that those little creatures create. Okay. So I'll do that, and then I'll go back again to the comments. So in order to prepare that, I'm using some dry yeast. You can also, what I'm going to do, there's this little ostracod still floating around here, sitting here. So I'm just going to take it up here again and put it back. I might need it again. Yeah, and goes on top here. I just move it on out to the side. And I also don't need this here. So I've got a little petri dish here, and what I usually do is I take some dry yeast. So you can also use fresh yeast, but because I don't need a lot of the yeast, I always use dry ones because it can be kept much better. The simple Saccharomyces cerevisiae, simple baker's yeast. And now there are a little tiny of these dry yeast grains here. Of course, I could place one directly on the on the slide, and then you can actually see the particles come off. Or there is also the possibility to first dissolve it in a little bit of water, because you have a little bit more control this way. And this way the cells of the yeast will come off. And then I'm going to take a little bit of the solution here, of the suspension, and add it to the Daphnia. Okay, and this takes again a couple of minutes, 2 or 3 minutes, and I'm going to use this time again to read a couple of the comments here. There's of course some communication going on inside the chat among the members here. I know because I've seen another YouTube 1,500 times using a 16 times objective. Yes, so you can get a total, you can change the magnification around by using different eyepieces and objectives. Okay, I recommend the Swift 380T. Yep. Have you watched the Journey to the Microcosmos and Science Fi? Yes, of course, I know those channels. I should always do the stream at this time. Well, I'm going to check a little bit on the statistics later on. Maybe I'm going to do 2 streams, one on Saturday, one on Sunday, but each one a little bit shorter, because I used to make the streams 1.5 hours, but this is a little bit too long for me as well. But maybe I was thinking maximum maybe 1 hour, and then I could actually think about doing 2 versions. Okay. What's this jiggly thing inside the flea? And these are the organs that are moving. So you're able to see some, yeah, the digestive systems. And I did not research this yet, but I think it could be maybe some breathing organs as well. Yeah. Okay. The objectives you're using are the objective colors are, yeah. Yes, a fold scope is cheap and have good quality. Well, the fold scope is a so-called a paper microscope. Is a single lens microscope. Is nice for yeah, for projects and so on, but it's not always very suitable for observing all specimens. So putting a water flea under the fold scope is not going to work well. It's going to be crushed. Okay, so this is a little bit yeah. Okay, and there are more. Can you show DNA the next live stream? Yes, I've been actually thinking about doing this as well. I have to tell you, DNA itself is too thin and too small to be directly seen under the microscope. However, it is possible to still see strings because many DNA molecules sticking together still make a string yeah, like structure. So you are able to see the long strings, but it's not individual DNA molecules, but many of them bundled together. Okay, but I can show you how to extract DNA. It's actually quite a straightforward process. It's actually surprisingly easy. Yeah. Can you give me a microscope because I'm very poor? Oh, that's a little bit difficult to do as well. It would have to be sent. But what you can do is the following. I'm going to show you something. The easiest thing that what you can try is the following. Okay, I got to bend down. Is I recommend that here I can actually show it to you here. Look, this is a lens. It's 10 times, and what you can do, and this actually does work, is you can put this on your mobile phone, and then you put a microscope slide over it. Yeah, it's like a microscope. Okay. As a matter of fact, yeah, I don't. That's actually also something I might want to do in a video to show how this works. It doesn't work quite as well, but it works surprisingly well. Okay, so you get one of those jewelries. So basically, people who are you know, looking at jewelry, for example, they use those. You put it directly in front of your eye. Yeah. So this is something I can also recommend. Yeah. So I'm writing my final biology thesis on paramecium. Okay, that's nice. Splitting the stream into Saturday and Sunday is a great idea. Allows more people to join and more themes to be covered. Yep. Okay. Yeah. We'll see how this works. Every stream is a little bit improvised, so the things that I'm doing right now with the yeast is not exactly the same thing that I've been yeah, doing yesterday, but it doesn't matter. Okay, I'm simply going to try it anyway, and we're going to see how it works. So what I'm going to do now is I'm going to yeah, show you how this works. You might not know this either. Yeah. I've got some pipettes here, and what you do is you go in here and you pick up a fresh tip. And yeah, it's now 15 microliters that I'm going to pull up using here. You just basically push it in and then you take up some of the liquid. I think this is going to be enough. Here is a little bit on here. And now I'm going to add it directly to the water flea. And you're going to see a lot of dots floating around. And these dots are individual yeast cells. Okay, so this will be now yeah, quite a mess. Okay, just watch this here. Okay, you can actually see it also in the corner. Yeah, I'm approaching and it's out. Yep. Okay, here we go. Okay, so all of those dots that you see are the yeast, are yeast cells. And yeah, I'm feeding it now. And now the yeast cells has the advantage that you are able to see the water stream now. And somebody just wrote that somebody's writing a thesis on paramecium. If you add a yeast to paramecia, then you're also able to see the water stream like this. So that's actually a very yeah, nice way of yeah, making things visible. Yeah, and yeah, and let's have a look at the ostracod. Maybe the ostracod is also doing. But this one is not moving so much anymore here. Okay, what about this one over here? Where is it? Sometimes it, yeah, here's another one. Okay, so I'm going to feed this also some yeast cells. Okay, not so much as the other one. Yeah, now it's moving. Okay, Let's go in a little bit. So, yep. Yeah. Now you're able to, with, yeah. Each movement here. Let's have a look at the larger one again. Here, that's the larger one. Yeah, you see how this guy is sucking all of the cells in here. Yeah. So you can use not only yeast cells, but you can also use a very small amount of milk because these are fat droplets. And the fat droplets can also be used to make the water movement visible. Okay, so again, also you can do this with ciliates, with paramecia, and so on as well. You add a little bit of diluted milk. Usually, if you add a small drop of milk, it's way too much. So I would highly recommend that you dilute it down first, and then you're able to also see the movement better. Sometimes it's easier to. Okay, yeah, here we go again. Yeah, it could be a little bit, let's add a little bit more water because it appears a little bit the water starts to evaporate already. So I'm going to add a little bit more of this water here. Also to give them a little bit more possibility for movement. Yep, here we go. Yeah. So it's creating this little vortex here, a little bit more maybe. Let's add some more of the. No, it's not taking anything in. Maybe it's gonna start again in a minute or so. And here we go again. Okay. Yeah. So maybe it does take a little bit of time to realize that there is some food here. Okay, and I think you can, yeah, oh, now just pooped again. So occasionally you're able to see, let me turn on the arrow here. Where's the arrow? Yeah. And so occasionally, this here is the digestive system, and just before, yeah, it was not full. Yeah, so it was actually half empty. It was only dark. It was dark only here in the back, and now even the front part is filled up again. It is dark. Yeah. Yeah, so you can actually see this way that the digestive system is slowly starting to fill. So if I were to transfer this now into maybe some water where there is no food, then this digestive system is going to start to clear and become clear, and then you're able to see it again. But what I've heard as well, what some people are doing, I've not tried this yet, but it is also possible to stain the yeast cells before feeding it, and then this way you're able to also give it some color. Yeah. Or I could imagine, I've not tried this yet, maybe by feeding it not yeast, but maybe some algae, single-celled algae. I could imagine maybe it starts to become green. Yeah. Because very often the organisms, they take up the color that of the food that they eat. Okay. So we're just gonna watch this again. This guy again a little bit. Yeah. And it seems to be already pretty full because you see all of the digestive system is full now, right? So it would actually be also quite interesting to see that whether it's taken up, it pooped again, to see whether it takes up food. Yeah, so maybe that's the reason it's starting to take up food now, because maybe there's again a little bit of space in the digestive system. So you see that the water starts to stream again. Okay, I'm going to go again over to the chat. Yeah, thank you, because inspiring. Put a laser lens on the mobile phone camera, simple and cheap. Yes, I'm doing bachelor's in microbiology. Any advice? Well, I did a master's degree in microbiology with a focus on molecular biology and bacteriology. Advice that I can give you? Oh my gosh, I mean, I don't know where to start here. Do that, where you learn a lot. Write a bachelor's thesis in those areas where you learn a lot yourself and which interests you. And don't forget one thing that, if you're already asking me, I've met a lot of people during university time who said this, well, I've got a degree in, I don't know, microbiology now, or in this area or in that area, and that's because my training, and therefore I want to do exactly the same thing that I've studied. And then this kind of limits you a lot. If you're studying biology or any science, you are trained as a scientist. Essentially, that's the important thing, is that you're able to think scientifically about the world, and then you can try to use this knowledge about science and about biology also in different areas. You can also go into journalism if you want to. You can go into education if you want to. You can go into, I don't know, development of laboratory equipment if you want to. You can go into sales. A lot of people at that time went into government office because for environmental protection, for example. So there are lots of things, and the advice that I can give you is don't limit yourself in your perspectives, but be open-minded, because whatever you learn, you can apply it in many cases to many different areas. Yeah. So, what is the most expensive microscope you have? I'm not going to talk about that. And because I think price is not it should not be a how do you say, a criterion here, because expensive microscopes do not necessarily automatically always solve provide that what you want. Sometimes cheaper microscopes are actually can get the job also done. I've actually met someone who wrote me an email several months ago, and he said is that he actually sold his expensive microscope again because he was so afraid of using it, and he got himself a cheaper one. Yeah. The journey to the microcosmos put your magnification in the videos, but yours you don't. Why is that? Yeah, that is correct. And I've made several videos about this as well. This is has several reasons. What it does not make sense. Let's put it this way: what magnification should I put here now on the screen? And this is a very, very common question, but I would like to ask you here now. Look at the. I'm using now a 4 times objective, but what is the magnification of this water flea? Is it 4 times? No, it's much larger than 4 times. Because there are 2 different types of magnifications out there. There is the so-called linear magnification, and there is something we call just magnification. And right now, when I look through the eyepieces, I get a 4 times. I've got 10 times eyepieces. 4 times 10 is 40 times. But the camera does not take the picture through the eyepieces, and the size on screen, or the actual magnification on screen, also depends on the size of your monitor. Because that's a different magnification. So whatever magnification I put on there is not going to be correct. And what Journey to the Microcosmos does is they include a scale bar. So the only thing that would really make a lot of sense would be to include a scale bar. I have done this as well. However, the problem here that I have then is that when I make my videos is sometimes I don't just take the video as it is, but I crop in. And then the scale bar is also not correct anymore. I have to crop in also on the scale bar, which is usually in the corner, which you don't see properly. So this means I'd have to do, with every clip that I edit, I'd have to do a recalculation of the scale bar. And that's a lot of effort. So for this reason, I don't include that. So over here, I wouldn't even know which magnification to include. The only thing that I could say is I'm using a 4 times objective. But it would look different in your case because your camera sensor might have a different size. So, and for this reason, I find it relatively difficult to include scale bar, especially magnifications. But I appreciate the question because this is one that has been posted quite often. Sometimes I've also included a scale bar. What does it really tell you? If you say, okay, that's I don't know, 10 micrometers. I mean, what's 10 micrometers, right? It's very difficult to also have a connection to real life. And I'm kind of solving the issue about the magnification by having this camera on the side here, so that at least people know which objective lens I'm using. So it's 4 times or 10 times, but But essentially, if you were to look at the water flea using a 10 times objective, you're probably going to see it differently because your field of view is different. So again, you see, it's somewhat a complex. It's a complex thing. I just got my microscope set up on the desk again a month ago or so. I just started experimenting with some plant cross sections. I wonder how the water fleas reproduce. I think some of them are parthenogenic. This means they are able to come out of unfertilized egg cells, and some of them are. And over here, what you see in the back, this actually is yeah, an egg cell. Where's the arrow here? It's actually only one. I've seen already some with many of them. Yeah, you can sometimes actually see them develop in there. Yeah. Looks like they increase their feeding movements when they actually detect the presence of food. Yes, this would actually make a lot of sense. Why invest energy if there's no food present? And I assume also that it depends a little bit on how full they are. Okay. So basically, how full is their digestive system? So that is the other one that we have. Okay. Yeah. So okay, here we go again. I wonder if the yeast would be clear in dark field. Yeah, I don't have a high magnification dark field here right now, but no, it won't be clear. You will actually see the cells bright on the dark background. Okay. Milk is actually even better in my opinion because no solid particles. Okay. I think the thing is the following, that those clumps that you see here, the solid yeast cells, they kind of yeah, stick to the microscope slide, and in milk, the fat droplets are actually moving much more freely. So yeah, so you can also try this with milk. Do water fleas have eyes? Yes, as a matter of fact. Let's zoom in. We can actually see them. Yeah, here. This dark spot that you see, that's the eye of the water flea. Yeah, and not only that, but actually it's connected to muscles, so it's able to move. Yeah, so they are able to detect brightness. I don't think that they have a very good resolution, but I can, yeah, in clarity. But yeah, they do have eyes. Yeah. Yeah. So let me go down again here. Give me a little bit of time. Can you put different organs under the microscope, like brain, liver, kidney, cardiac, etc.? Yes, I can do that. You know what? Why not? I can prepare maybe in an upcoming live stream. I could actually prepare some ready-made slides. Okay, so I've got some slides of different organs. Yeah. What do you think of amoeba? Yeah, I made some amoeba videos. Yeah. They're fine. They're interesting. So if you check this channel, I've actually published a couple of videos with amoeba. Yeah. You should try putting unused or used toothpaste under the microscope. Yeah, or more interesting yet, maybe mix some toothpaste with some microorganisms and see what happens. Okay, maybe is the toothpaste able to kind of, I don't know, attack some microorganisms? Because toothpaste, they have sometimes those substances in there, like how do you call this? Peppermint taste, and all these oils yeah, basically sometimes also have disinfecting properties. I have a love-hate relationship with bacteria. They're cool, but they have ruined many cultures and experiments of mine through contamination. Yeah, says someone who's been working in bacteriology. So essentially, this is one of the things I can reassure you. You are not the first one with that problem. It goes back. The problem of contamination goes all the way back to the 19th century, when people were brewing, making wine and beer, for example. And it was in France at that time, Louis Pasteur, a very important French scientist. He was doing a lot of fermentation experiments, and he discovered that actually it is yeasts that cause fermentation to happen, and that bacteria contaminate. If bacteria present, then it spoils the whole thing. Yeah, and yeah, so he was Louis Pasteur was not only important for discovering pasteurization, which is basically killing of bacteria and making food storable by heating it up. But he was also very much involved in brewing, and at that time many people had problems with contaminations who were making wine and beer, and he actually improved this quite a bit, and he was able to discover it was the contaminations, and this was of course of highest economic importance as well. Yeah, microbes do not have liver, kidney, heart, and brain. That is correct, but they have so-called cell organelles, which are subcellular structures, yeah, that basically are able to perform some of these functions. Okay. If you want some mammalian tissue to observe, it's probably best to go for farm animals. You can get it from your butchers, probably. Yeah. What I've done already as well, I can also show you. It's quite easily possible to put meat, the muscle fibers, under the microscope, because you're able to see the so-called striations, of the striated muscle, which is also quite interesting. So, thank you for the recommendation. I'll prepare a video for how do you say, observing mammalian muscle tissue, and not only that, generally mammalian tissue. How long did it prepare this slide here? You're talking about this slide here? The preparation took me no time. I just took out the water flea and put it on the microscope slide. So there is no preparation here. Yeah. Okay. So quickly, permanent slides are boring. It looks like you're not just watching a picture. I prefer making live ones. This is an interesting comment. Permanent slides indeed have no movement. That's correct. And therefore, usually during those live streams, I do always use, I try to use, if possible, live specimens, because there's some movement going on and it's simply more engaging. Yeah. So yeah. However, there are simply some specimens where you have to make a permanent slide. Otherwise, you're not able to see it. So I'm going to add a little bit more of the yeast solution here. Okay. Yeah, and the guy's already pretty full, I guess. Yeah. Look, the whole digestive system is already full. Okay. But what I'm going to do now is the following. Maybe I'm going to try exactly the opposite. I'm going to now take it off again and put it on a clean slide with some clean water. And maybe we're able to see over time how the digestive system starts to empty. Okay, so I have to carefully take it off. Okay, so let's take the slide here. Okay, so where is it? I'm going to take it up here. Yeah, no, not enough. Here we go. Here it is. And I'm going to now remove any excess water here, and I'm going to add a little bit of more water. I need a little container for water. I'm going to use this Petri dish here. And let's simply add a little bit more, just some. So I'm going to remove first. I'm going to use a larger pipette now. Okay, so let's remove all of the excess water here with all of the yeast first. Put it here. And I'm going to now wash everything. There is always a change of water. There's always a little bit of an osmotic stress. Okay, for the organism. Not all organisms like it. If you change the water, this now is tap water. However, the tap water that we're using here is does not contain chlorine, so it's going to be fine, I hope. So, and now let's put this one over here now again under the microscope. So with all of the food removed, and usually this is what I do in order to make nicer pictures, because it's simply see, and now we can actually watch it poop. And maybe over the time we're able to see how the digestive system starts to clear again, and then maybe we'll see how far we're able to take it, and then we can add some food again. But usually every I don't know, once every minute or so it poops. I've been observing this also some time, but let's just leave it like this. You know what? If you want to have a little bit of color, let's add a little bit of color so that it looks a little bit nicer. Okay. This is an osmotic stress for the organism, and it usually takes a little bit of time for it to get used to. Is it possible to trigger apoptosis under the microscope and be able to observe it? Apoptosis is programmed cell death. So I'm quite sure that somehow it should be possible if you have, for example, cell culture cells, so-called HeLa cells, and if you add certain substances, I'm quite sure that you might be able to trigger this as well. I've not observed it yet. Okay. What should I do if there are no good ponds near me? You're going to be surprised. If you want to observe these, what you do, water crustaceans, especially, then you can do the following. Number one, you can buy them. Some shops sell them as food for fish, for aquariums. And or what I would actually then recommend is if you want to observe saltwater crustaceans, buy so-called sea monkeys. They're called sea monkeys. Obviously, they're not monkeys, but they're so-called brine shrimp, Artemia, and they are sold as eggs together with salt. And all you do is you put it the mixture into some water, and then they're going to hatch. Yeah, they will actually grow much bigger than those, and they are sold for hobby purposes for kids. And also, to make your own fish food if you have an aquarium. So go for so-called sea monkeys, and you can buy them online, Amazon, and so on. And if you want to know how this actually looks like and how it works, I actually some time ago did make live streams and also YouTube videos on this. Yeah, so it's quite if you're interested in doing that, then I would actually go in that direction. Are you able to dye the flea, so to stain it? Answer is yes and no. Any type of dyes that you use will most likely harm the organism. But what you can do is you can use food that is dyed. So some people, as I mentioned already, have dyed the cells, the yeast cells, and then you should be able to also be able to see that the digestive system in a different color. Okay. I live in Austria. That is correct. Okay. Do you know where I can buy some bacteria prepared slice in Europe? I'm in from Denmark. About bacteria, you see, there are. If you go, what you can do? Yes, I do know. If you want to, they're not cheap. Okay. I'm going to type it into the comments section now. The address. So prepared slides. This is a prepared slides. Leader.de. I'm going to post this now in the comments. I'm not affiliated with them, but this is a huge a company that sells a huge number of slides, and you can also buy them individually. They're not cheap, but they're of high quality because they're preparing those slides also for medical research and for medical training. Okay, leader.de is and you can download the PDF catalog from them, and it's a huge catalog. Okay. However, if you want to observe bacteria, my recommendation is don't grow them yourself for safety reasons, but maybe you just want to buy yourself some yogurt bacteria and make some yogurt and put the yogurt under the microscope. Okay. So this is something that I recommend. You can find biofilms. Yes, that's correct. Okay. There are no fountains or parks near me, but I can observe a specimen. I kind of want to observe living. What you do is if you don't have anything, you make yourself a jar with water, and maybe you can keep your own jar somewhere. You see how it's slowly starting to poop out the things here. Look, I wonder if these are actually yeast cells that it's kind of pooping out here. Yeah, all these things here on the side. Yeah. You could even try puddles on your gutters in your house, but it's usually the puddles have to be a little bit old. Okay, so if it's just raining and there is a puddle that's been standing there for a day or so, you're not going to find a lot of interesting things. If you don't have a good exactly, put some plant material in there. A video demonstrating osmosis. Yes, I could do that. And as a matter of fact, I've recently published a video about osmosis. And if you check the channel, just recently I made some videos about this where when you add salt water to blood, for example, then you can actually see the red blood cells shrink. With onion cells, I've done this as well. I've also added salt water to paramecia. They will also lose water and die. Yeah? So this is yeah, something that I could also demonstrate in one of the live streams. Yeah? So it's even smallest life is beautiful to watch. Yes. Okay. So is there, okay, so these are all of the comments right now. Okay, so what we're going to do now is we're just going to zoom in a little bit more. I always have to check. It's already 44 minutes I'm talking here. I wonder how many, I mean, there are lots of comments anyway, so this time seems to be also quite well possible for some people. Yeah, also something that you might be able to see here, if you look at on the surface here, you see that there are, it looks almost a little bit like Vorticella or some other microorganisms growing on the yeah on the water flea, yeah, as well. Yeah. It says it's a scam. What is a scam? Okay, I don't know. Yeah. There are microbes in salt water, but how do they survive in that salinity? I mean, osmosis does not affect them. Okay, I can tell you that. They have found bacteria also in the Dead Sea, which is actually concentrated salt water. And so how in the world is it possible that they survive there? And I will explain to you how this works. What they do is first of all, the problem is if you put cells, microorganisms or so, into salt water, because of osmosis, they will lose water. Okay, this is also one of the, and if you put cells into, let's say, distilled water, which does not contain any salts, in many cases, the cells will start to swell and then they might pop. This was actually one of my worries here with the water flea, that when I transferred it into tap water, it's not distilled water, tap water, that still this would cause too much water to go into the cells of the water flea. Now, what about cells that are, for example, living in the Dead Sea? Normally, they would lose a lot of water by osmosis, and then they would start to shrink. However, this does not happen there, and the reason is because they have a mechanism where they actively pump salt into the cell by active transport, so that the inside of the cell is more salty than the outside of the cell, even though the outside of the cell is actually already saturated. I don't know if you're able to, if this makes sense. And this basically means that the bacteria living in the Dead Sea, there will be actually even water going into the cells of those microbes, and they will not dry up because the inside of the cell is saltier than the outside of the cell. So yeah, so this was a short answer to Jester, who was asking about the salinity. I hope this was clear. Yeah. It says LIDA has an outdated certificate. Ah, interesting. No, actually, I know that the website of LIDA is extremely old, but the microscope slides that we are using in school are actually from them. Yeah, and they have a pretty nice large PDF. Yeah, so maybe they have not been updating the web page. Yeah, okay. Their certificate has expired and the web page apparently taken by another. Really? Let me quickly check this. This would surprise me because I recently checked. This is weird. Indeed. It's, yeah, correct. The web page of LIDA does not work. This is strange. No, I can confirm that they have some issues. But yeah, I checked several weeks ago and it still worked. And actually, I had a look at the catalog because they had a nice catalog, also in English, of course, where things could be ordered. The webpage was a little bit old, so maybe, I don't know, maybe they're updating the webpage. Okay, yeah. So. I've been playing with stains recently. I thought Tolite Blue was capable of staining nuclei, but they're not visible when I use it. I was incorrect, but how about staining organelles? It depends on the organelle that you want to stain. Generally, the stain that I generally recommend most of the time is methylene blue, also known as Löffler's solution. This is a not concentrated methylene blue, but it stains DNA and the nuclei quite well. So it's a general purpose stain, but like in many cases, it might actually kill the organisms that you look at. Yeah? Have you streamed on Twitch before? No, I have not tried Twitch yet. Okay, and I actually was briefly thinking about it on Twitch, but then I decided not to do that because otherwise I'm going over too many different platforms. Okay, have you ever broken a microscope accidentally? Yes, I have. Yeah, it was an old used microscope. I tried to fix it because and I couldn't put it back together again. I didn't have the proper tools. It was the ball bearing system of the mechanical stage. It was horribly difficult to fix. Another microscope I broke, and this was a never ever do that. I will tell. Okay, I'll tell you. This was just plain stupid. What I've done is I have the coarse focus knob of the microscope. It was a very old one, but I tried to adjust the focus tension by rotating the focus knobs against each other. In some microscopes, you can do that. You can adjust the tension of focusing by rotating the knobs against each other, but not this one. And that ended up breaking the axle. Yeah. So this is something you should never do. Never ever rotate the knobs against each other. But in one stereo microscope, you were supposed to do that, and it worked. And I basically had a blank out, and I tried it also on another microscope, and then I broke it. Yeah, so that's it. So it's something that you shouldn't do. Yeah, it can happen, of course. Also, a second thing that once happened to me is that I accidentally rotated one of the objectives into mounting medium. I was too sloppy, and I quickly had to clean it off using alcohol. This was a close call because you don't want to get mounting medium, which really turns hard on your objectives. Yeah. Is water fleas visible by the naked eye? Yes, of course. Look, I'm going to show you. Desk. Do you see those dots here? Yeah, these are the Ostracods and water fleas. Yeah, they're about 1 millimeter or 2 millimeters in length. So very easy to see without, yeah. That's the reason why I'm using the lowest magnification here, because they're easily visible. Yeah. How scientists determine that some things made up, that many cells like this water is made up of thousands of cells, but how do they count it? Yes, that's a good one. Indeed, I also wanted to figure this out. There was a scientist who actually has done this, and what he's done, apparently he somehow stained the nuclei, and he somehow counted this, and he discovered that certain organisms, like for example rotifers, have a constant number of cells. So there are certain organisms, humans for example, mammals, they have a different number of cells. So you and I have a different number of cells, but there are certain organisms that have always a fixed number of cells. And rotifers belong to those, and he actually, they actually counted the cells. Maybe it's possible to separate the cells somehow. Using certain chemicals, or to do staining of a nuclei, and then you have to count the nuclei. And they discovered that rotifers, for example, contain around 1,000 cells. Okay. Where did you get that microtome from? The sections of the asparagus? I'm still doing it by hand and want a proper tool. Okay. Here it is. Luckily, I've got it here. Honest, I don't know where I bought it. I bought it somewhere either on eBay or on Amazon. It's not the highest quality microtome. Yeah, it looks like this. Okay, this is again a bad image. Yeah, it looks like this. There is a glass plate here. And when you buy a microtome, just be aware that there are different kinds. There are those that have a clamp in here, where you can open it up and where you can clamp it closed. And then there are those that don't. And if you have already the choice, get those with a clamp. Okay? Otherwise, it's going to be really difficult for you to fix the specimen in here. Yeah, it came also with a second glass plate, which I never used. Yeah, the second glass plate still packaged, and it came up. It came with a knife, which is so bad that I've never used it. It's a disaster. So we're actually making the cuts. What I've done is I've used some razor blades. Yeah, these are razor blades, which are very cheap. Okay, but very dangerous. Yeah, so you have to be really careful. Just show you. I need to go down here. Where is it? Yeah, those razor blades, they come in little boxes like this, okay? And you have to move it out, and then packaged in here, you know, you've got the razor blade. Yeah, yeah, be careful with this, because yeah, if you hold it, you might actually cut yourself. Yeah? So, but I found those razor blades to be much better than, for example, those cutting board knives. They also kind of work, but they're not quite as sharp. Yeah, so, yeah. The problem with the razor blade is that they're flexible and they bend. When you cut it, they bend and you don't get an even cut. And for this reason, that's also one of the reasons why I 3D printed myself. And there are razor blades also around that have an edge that are a little bit more solid. So you have to kind of think a little bit of how can you actually make sure that it's kind of stays flat. And the correct angle is very important as well. Yeah. So yeah, so this was a short microtome question. Let's go back again to the microscope view here. And as you can see, and that's actually what I wanted to show, where is the arrow? Yeah. Can you see over here, the top part of the digestive system is slowly starting to become empty. And as it's kind of pooping out all of the rest here. Yeah. So, yeah, here again. Yeah. Let's, yeah, so. I just wanted to, yeah, demonstrate this to you. Where else? Okay. What's your heaviest and biggest microscope? I think that's the one that I have standing next to me here. Yeah, it's the Olympus microscope that I have, and it's also kind of heavy because I've got a digital SLR camera on top of it. Yeah, yeah. Can you use again this time slot in your next live streams? So you know what I'm gonna do probably is the following. I might split up the live stream into 2 shorter live streams. So maybe I'm gonna do one on Saturday evening, my time, and maybe one on Sunday morning, also my time. Yeah. Why is the background colored blue and orange? Because I've got a certain contrasting technique called DIC, which uses polarized light. And yeah, I'm just using this to make it look nicer, colorful. My favorite type of light microscope, I don't have a favorite one. Well, I mean, that's the one I'm using maybe right now, isn't it? Yeah. There are 2 types. There are compound microscopes and there are stereo microscopes. And I use both of them, but I probably use more the compound microscopes. But yeah. Cutting myself with the blades, yeah, is very dangerous. Yeah, I don't like the thing about science. It told us he discovered that, he invented that, but they don't really tell us how. Even they told it's a little they told us he. Okay, I guess Jester has a question about the following. I guess about how things were discovered. I have a book recommendation. I highly recommend the book, and the name of the book is a little bit similar to the name of this channel. The name is by Paul de Kuif, and the book is called Microbehunters. It's a 1927 book, Microbehunters. It's still sold as an ebook and also in printed, and it is a very, very readable, very, very interesting and also easy to read book about the early discoveries in microscopy and microbiology. So I highly recommend it. Microbehunters, 2 words, and you can buy it over Amazon. And the thing is that it explains very well how the first microscopists made discoveries, how were certain diseases discovered. And simply by reading this, you also understand a little bit some of the methods that they use. Don't forget that those early folks, they had no idea. They were just inventing pretty much everything. At that time, they didn't even know that cells divide and that they can reproduce by dividing. So there are very lots of interesting stories in there, and I can highly recommend that. Okay, do cells have a chemical formula? The answer short is that cells are made of parts, different parts that are chemicals, and they do have chemical formulas. In very brief, cells are made of carbohydrates, lipids, proteins, and nucleic acids, and they basically, yeah, have a certain structure. Yeah, so there are certain chemical components that you can find in all living things, and this is known what they are. What's the smallest thing besides bacteria you could manage to see under the microscope? This is an interesting one. If you use dark field, you are able to also see structures that are significantly smaller than what the microscope is able to resolve, because you're just seeing a bright spot. Okay, so you see that something is there, but you're not able to actually see the structure of it. Yeah, it could be that you're definitely able to see certain subcellular structures, definitely, but only if the contrast is high enough, and this would be in the case if you, for example, use dark field microscopy. Have you ever gotten angry while using a microscope? You won't believe it. Yes. Okay, when I basically made a stupid mistake and when I rotated one of my objectives into mounting medium. Yeah, I got angry with myself. I said, come on, how can you make a mistake like this? I'm not talking about immersion oil here. I'm talking about mounting medium, which actually turns hard. Yeah, I immediately, I was simply sloppy. I was not patient enough. I did not wait for the mounting medium to dry. And I simply rotated it. Great. I had to immediately take out the objective. I had to clean it carefully. Everything was fine. Yeah, but this was an experience. Can water fleas become constipated? I don't know, but I would say probably not, because unless maybe the food solidifies that it takes in. I would say probably not, because the food particles are too small. Kind of chemical formula are for single chemicals, and cells contain many thousands of chemicals. Yes. Okay. People, we are now 1 hour live, and I promised myself not to do live streams too long. So yeah, basically, if you want to see a little bit of what I've done yesterday, the live stream of yesterday where I added some caffeine and alcohol to those water fleas is already available. Today I did a little bit of feeding, and there were lots of questions again, which is very nice, which actually shows that people join in the live stream also today. I'll be looking at the statistic, and I'll be thinking about a little bit how to make it in such a way that people all around the world are able to be able to watch it. What I'm going to do now is I'm going to leave it at that. I just want to inform you that next week, Saturday, Sunday, there will not be a live stream because I have an appointment, some time conflict. And that's why it's not possible. But then afterwards again, there are plenty of videos that I've got on my a YouTube channel. So if you are interested, please go back a little bit in time and look at some of the earlier videos as well. I do also have a second channel called Microbehunter Microscopy, where I talk about more about microscope and microscope reviews and questions and so on. So there's, I'm quite sure, plenty of things for you to observe, or you can watch one of the other live streams, the previous ones. Yeah, I'm going to say bye-bye now. Okay. I wish you all the best. Hope that you liked it. Do leave your comments again. Either in the chat and or in the comments section. I wish you all the best. Happy microbe hunting as always, and see you around. Bye bye.